1985 is the 25th year since the founding of the American Society for Cell Biology. My name is Mary Bonville, and I'm one of the more than 5,000 investigators and students who will be attending the Society's annual meeting this year. In celebration of that anniversary, I'd like to review the development of the field of cell biology by following the career of just one of its distinguished pioneers. That pioneer, Keith Roberts Porter, was born in Yarmouth, Nova Scotia in 1912. After graduating from Acadia University, he left that Canadian province and went to Harvard as a graduate student, completing his PhD there in 1938. In the nearly 5 decades he has devoted to research since then, he has inspired many of his contemporaries and younger colleagues, all the while contributing to our rapidly growing knowledge of cell structure and function. In 1939, Porter was a postdoctoral fellow at Princeton, and like many postdocs today, he was looking for a job. His interest in developmental biology led him to write a letter to Dr. James B. Murphy of the Rockefeller Institute for Medical Research in New York City. To his great surprise, Dr. Murphy read his letter and offered him a position. At Rockefeller, Porter learned to do tissue culture and then began collaborating with Dr. Albert Claude, who was a very active member of Murphy's lab.
Claude had published a paper in Science, I think, in which he described the microsome, his isolation of it, and so on, and how similar it was to the microsome isolated from tumors. There was this similarity, tumor activity and inducing activity on one hand and none on the other. But he'd characterized these things and published the results. And a man downtown working at Interchemical Corporation, this laboratory had the only electron microscope in New York. And he read this article in Science, and he called up Claude and asked him if he'd like to come down and look at his microsomes in the electron microscope, because you couldn't see them very well into the light microscope, just in dark field, you know. So Claude had established a contact with the laboratory at Interchemical and with Ernie Fulham, who was the microscopist. And eventually, out of curiosity, I went down and got acquainted with those people as well. And I began to learn what was required of an object to be useful in electron microscopy, notably that it had to be very thin and that it had to be dried, so on. We were both, Claude and I, both interested in seeing whether we could see cells in the thing, and the cultured cell turned out to be a fairly good object.
Well, when did you get the first image?
Late in 1944. I succeeded in getting, I believe, 2 cells on a EM grid, and one of them was stretched out over an opening, which was a great, a great good fortune. And we stayed all night. We took pictures, as many as we could, and they turned out to be quite interesting.
Was it difficult for you to interpret?
Sure, because we were seeing things for the first time, and we recognized that, and there was no name for it, and there was immediately no recognition of what it might be. In fact, we didn't know enough cell biology to know that the basophilic component had been described about 30 or 40 years before that. So immediately, there was no immediate decision on our side to say, so this is the basophilic component everybody else has been looking at. Nothing of the sort emerged. All we could say is that we were looking at a reticular structure, and out of that eventually grew the name, and it was obvious it was not part of the cell surface immediately under the surface. It wasn't an ectoplasmic component, it was an endoplasmic component.
In 1948, when Claude and Porter, together with Ernest Fulham, were beginning to publish their discoveries, the new findings excited the imagination of a young medical doctor from Romania who came to Rockefeller and joined the lab. Dr. George Pilati reminisces about that remarkable institution.
When I came there, it was very clear that there were lots of attractive features. One was that total dedication of the people to their work, their deep involvement in the work. The other point was that there was a system of communications between the different people in the laboratory and all the laboratories at the university, at the institute. At that time, a rather small outfit. One element was a system of seminars attended by practically everybody, including the director and the administrators. Yeah. And the other thing was a luncheon taken in common, a famous dining room, which was ornate, elegant, a little bit, let's say, excessive in terms of comfort and decoration, but in which a lot of interchange was taking place.
In 1949, Dr. Murphy died, and Albert Claude returned to his native Belgium. The director of Rockefeller, Dr. Herbert Gasser, took Porter and Pilati under his wing.
Gasser made it easy for us in many ways. He was patient, he was considerate, he was interested in what we were doing, and he knew what we were doing.
A successful research institute is based on the formula mentioned by Keith Porter. You find good people, you give them the resources, and let them do their work without administrative guidance or interference. Yeah. He modified it to some extent, because instead of disbanding laboratories, as the disappearance of the head of the laboratory, he allowed the people, if they are promising, were promising and interesting, to continue. And in fact, the most successful part of his administration was connected with this kind of policy, which allowed people like Stan Moore and Bill Stein and Lyman Craig and the two of us to continue under a form or another our work without having really come to the level at which in the past it was considered acceptable for us to become independent.
Gasser also shared the enthusiasm for using the new instrument and decided to purchase an electron microscope for Porter and Pilati.
You should realize that all the bills were paid through his office from the endowment. There were no government grants. The Institute was totally independent of that sort of support. And when it came time to get an electron microscope, he struggled with that cost of something like $12,000 for an EMU2, RCA EMU2, I think it was called. And there was a lot of opposition from the director of the hospital at that time to investing that kind of money, but Gasser took the bold step. Up until then, we'd been supported, or had been allowed to use the instrument that belonged to the Rockefeller Foundation, whose director was at that moment, was a very imaginative man who could see that this was an instrument worth having. Gasser took heart, I suppose, from that and finally invested in one for us.
With a lab of their own, Porter and Pilati started working to solve the technical problems involved in obtaining satisfactory images of cells with the electron microscope.
Well, I'd like to say something about sectioning, because it was the real barrier to rapid progress in collecting information at the level of resolution provided by the EM. You could work on cultured cells to a certain extent, but the culturing techniques were terrible then. Nowadays it would be simple, it is simple. But then it was a real problem. And there weren't many cells that you could get to grow in culture. And if you did grow, you're not interested solely in growing cells. You're interested in differentiating cells, and cells doing other things, functioning as differentiated cells. So the compulsion to produce a microtome would work became greater and greater, and several people took a crack at it. I was one of them, and I had just been in the lab of Bennett's out in Seattle, and he had made a monstrous microtome, which he said was necessary. It had to have mass. He'd calculated it all out. It had to have mass. It weighed about a ton. And it operated on the principle of bending a bar. He never published it. He never did anything with it. In fact, it's probably resting somewhere in some storeroom out there. But I came back and with the understanding with him that I would try to get our machine shop interested, and I did. But initially we made one, almost made it in the laboratory. We bent the bar, but not a massive thing. It was a simple bar. We put a lamp over it, gooseneck lamp, and it provided the heat for the expansion of the bar, and we got sections. They were beautiful sections. Gold, silver, and purple. And they were thin enough. And then Joe Bloom entered the picture one day when he was in the lab, and he said, "What the hell's that, you know? We can't have an instrument around here that involves bending a bar. That's not..." So he produced the elaborations like the gimbal ending of the bar, which gave it universal motion. And I said it wouldn't work because it had oil films. He said, "We'll make those junctions so tight that no oil will exist in them." And he put them all under spring tension. So that's how the microtome evolved. And that was really the biggest event in that year in electron microscopy, because it was available to people, and it just took off. I mean, everybody was cranking out sections. Literally. I mean, every lab was this way as fast as they could. And by 1959, that was 53, by 1959, we had all of, in papers or in publication, all of the basic information about the organization of important tissues and cells. It's amazing.
Palade recalls this exciting period.
Then there were improvements in fixation procedures. And then there were improvements generated in our laboratory. Then there were improvements in embedding procedures, which were coming from outside. But somehow we succeeded in putting all this technology together and producing constantly good results.
The resolution of the electron microscope led to many provocative revelations regarding cell structure. Biologists were learning to use an instrument that had previously been exploited primarily by physicists. These structural studies inspired Palade to ask questions about the function of the newly discovered organelles, especially the small particulate component of the cytoplasm.
From the very beginning, I started working with the idea that what has been done for mitochondria, particles which can be seen in the light microscope, by isolating them, characterizing them, etc., can be done now for the other components which can be seen only by electron microscope. By electron microscopy, like, let's say, elements of the endoplasmic reticulum, ribosomes, etc.
Peter Satir and Lee Peachy pay tribute to the contributions made by Porter and Palade in establishing the new field.
That Palade was one of the people who said, okay, now we've defined the organelles in terms of structure. Now they must also have a biochemistry. And he was the link between these two schools, if you like, and that is part of his genius, that he recognized that.
And Siekowitz was an important part of that as well.
And Siekowitz was a key, I think, to that, because Siekowitz was the person who gave the biochemistry its legitimacy, because he was a certified biochemist.
And then we started a very long period of collaboration, which lasted a good number of years, and which actually led to this idea that morphological structural studies must be integrated, or should be integrated, with biochemical and functional studies. And this approach, which was actually not unique with us, unique to our laboratory at Rockefeller, but was also applied to different extents in other laboratories, became the basis of the cell biology of today.
In the early 1950s, cell biology was becoming a recognized discipline, and it became apparent that a new journal should be devoted to the subject. Keith Porter tells us how the Journal of Cell Biology came into being.
It's so simple that it's easy to tell. Fawcett and I had prepared a manuscript on cilia, which after all was a fairly important paper, and it was good paper. Fawcett had written most of it. I'd done some of it. He had deleted some that I did, which was characteristic. And I presented it to the Journal of Experimental Medicine, and that was published out of Rockefeller. Very good journal, good distribution. But the manuscript did not meet with the approval of the editors. And Dole, what was his first name? Vince Dole, yes. Returned it to me with apologies, said that the editors had decided they did not want to publish that kind of paper. And he says, it's not that it isn't a good paper, it's just that the subject matter is not for the readers of the Journal of Experimental Medicine. And so I went back, started back to the lab with my tail between my legs, and met Gasser. And in a voice that I would attempt to repeat, he said he'd heard that the manuscript was being rejected. He says, that's ridiculous. So he said, why don't you publish your own journal? Why don't you start a journal? Just like that. And added that I'll find some money to support it. That's all it took. So that was the story. That's it. It was a very logical thing to do. I don't know why we didn't think of it, except it seemed like such a big decision. It's not the sort of thing you do every day, to start a journal. But it worked out very well.
At about the time that cell biology was being recognized as a field of study, Dr. Detlev Brandt became president of Rockefeller and opened its doors to a younger generation. Dr. Pallotti recalls that time.
To some extent, the Institute was operating as a relatively independent unit of the rest of the research and academic environment. So perhaps it deserves the title of an ivory tower. So what Brandt did was to open the gates of the ivory tower, taking advantage that the construction he inherited was solid and solvent. Okay? And within this program of opening the gates was the introduction of the graduate student program.
Lee Peachy and Peter Satir tell how it felt to be a student in that lab.
Yes, I think my goals and standards just leapt upward when I went to that lab. It was a new world for me, a new, and I'm sure that it wasn't an awareness. I sat there and said, I guess I'll scale my goals up. But there's no question in retrospect that my goals went up by a very large amount when I went to that lab. I was just in an environment where the work that was going on was just more important, more exciting. People were working, not so much working harder, but they were somehow keener. And I think there was a big change in my goals. I doubt very much if I would be where I am and where I've gotten today had I not gone through that lab or a similar lab.
I think everybody who went realized that there was something special going on, and it's an atmosphere and a time that's very hard to recapture. You know, a lot of the learning in the laboratory took place at tea time, where everyone would congregate and share. What I remember specifically is sharing the micrographs. You would bring in your pile of pictures from that day, and everyone would, either Porter or Pallotti or both of them would be standing there holding the micrographs, and everyone would be looking over their shoulders to see what features of the micrographs they thought were extremely important and new and you'd go away and say, oh yes, now this is fascinating. Why didn't I go and do that?
The late 1950s saw the growth of Rockefeller, paralleling the expansion of scientific research in the United States and abroad. Rockefeller also became a university, and its graduates included students in the new field of cell biology. This institutionalization of cell biology was acknowledged by the founding of the American Society for Cell Biology. Keith Porter tells of the society's beginnings.
That grew out of a study section, really, in Washington and Bethesda, when there was a general recognition by me and by other people that cell biology was a field expanding rapidly, and I even wanted them to set up an institute at NIH, Institute for Cell Biology, and I said I thought it should be an institute that was scattered over the country, and the laboratories would appear on various campuses of the country. And that was vetoed by the establishment at NIH that didn't want their laboratories dispersed. And so as a sop for my hurt feelings, I think they decided that they'd make some fairly large grants to a number of laboratories for cell biology, and that they would listen with favor to the establishment of a society. And they held true to their promise. We got together a representative group, including Paul Weiss and George Pilate, Fawcett, some Harvard people, of course, and Yale, influential folk. And we had a board, met a few times, and decided how we'd organize it. We had the first meeting in Chicago, as I recall.
During these years and the decade following, Keith Porter turned his attention to a number of problems, particularly the study of microtubules.
So it was not until then I went to Harvard that, and Sabatini and company published the paper on glutaraldehyde, that we took an interest in microtubules. And by then we had discovered that glutaraldehyde could be used quite successfully in fixing plant cells. It was better than permanganate and better than osmium. And Ledbetter, who was in the lab then at Harvard, was doing quite a bit of studies on plant cells and began to see microtubules in plant cells. And that was exciting because their distribution coincided with the deposition of cellulose in the wall, secondary walls of plant cells, where it is deposited in a pattern, you see. All cells had microtubules. And then we discovered that they were very common in the mitotic spindle and seemed to be connected to the chromosomes. They were non-randomly distributed in cells. And this meant that there was an organization which controlled their distribution. We went on to show that they had some relation in morphogenesis to the shape changing. Tilney entered the laboratory and picked up the thread and did the work on the axonemes of Echinosphaerium, the radiolarian.
In 1970, Keith Porter moved to Colorado to establish a new biology department in a new building that was to house the million-volt Electron microscope. The installation of the instrument demanded solutions to a number of engineering problems. Thanks to the devoted work of Dr. Mircea Fotino, a convert from nuclear physics, the facility was ready to be dedicated early in 1973. How did you decide that the time was right to begin using the high-voltage scope in your research?
I don't think that I ever asked myself whether the time was ripe for high-voltage electron microscopy. It has become obvious since then that the time was not ripe, that the high-voltage microscope was ahead of its time. but it was something that I wanted to try because I wanted to see the three-dimensional structure, especially the cytoplasm, because I felt that there must be something there that we didn't know about.
Perhaps you'd like to talk a little bit about what has come out of the laboratory from your own work and from that of other people.
Well, it is, I'm sure now, become evident to the world of cell biology, at least, that the ability to examine thick preparations has its advantages. The thin section is fine for some things, but when you want to investigate the continuity of a structure in a cell, a very small structure perhaps, it's important to see it in depth, see it in 3 dimensions. Lee Peachy has made good use of the high-voltage microscope in following the distribution and continuities of membrane-limited structures in muscle, the sarcoplasmic reticulum and the transverse tubule system. I think probably his images and his ability to make these available to an audience has done as much, if not more, than anything else that has been done with the microscope and by popularizing it. What we've done is gone in the other direction to look at the finer, smaller inclusions of cells, but also to get away from the interference of the embedding matrix and the artifacts that might be introduced by staining. Besides, we're looking at structures which have, as far as I know, no selective stain. We were exploring the cytoplasmic matrix with these techniques and finding things that no one else had ever seen. And we had the problem of proving to our public that we were looking at real structures, real in the sense of being there in the living cell and not produced by fixation or drying or even by the microscope. And we've succeeded in doing that. What we're looking at is a gel. We've known that the cytoplasm was a gel for the last, ever since before World War I in the 30s. Everybody talked about it being a thixotropic gel. If you shake it fast like that, it becomes a sol, but immediately it becomes a gel again. Well, a gel always has, biological gels have a set morphology, strands with inter-strand spaces filled with water, or a water-rich solution, something. That is a gel. That's what we're looking at. Now, the next question is, does that gel have information in space? Is it the same in one corner as it is in another corner? Or is it structured in such a way that certain things happen in one location and others happen in another location? That is the significant question, and that is what we're now working on.
Do you think you're convincing people to accept the microtrabecular lattice now?
It has been controversial. Everything new. The endoplasmic reticulum. You should have been around there in the first 7 or 8 years we were looking at that thing in cells. A number of people who came by to tell us it was just a product of fixation, osmosis, or something. It wasn't there, really. Of course it was there. We knew we were looking at a membrane-limited structure that was vacuolar in character, finely divided, distributed non-randomly, had structure. We knew all those things. We didn't know what it was doing. It waited for Palade and Sikovits to collect information on that.
What do you think about the microtrabecular lattice? Do you think he's going to be right about that?
Well, if I was a betting man, if I were a betting man, I would say that he is going to be right. In fact, there's no question in my mind. He's right in some respect. There is something there. It's not structureless. The space between the organelles is not structureless. There's something there.
Actually, it's the latest chapter in a search for the ground substance of the cytoplasm that he began essentially in 1945 when he took the first electron micrographs, because continuously he's been looking for what's in the space that nobody knows what's in. And first it was the endoplasmic reticulum filling the space, and then there had to be stuff between the endoplasmic reticulum, and then it was the cytoskeleton filling that space, and then there still were interstices, and so now you have the microtrabecular lattice.
Regardless of the controversy surrounding the microtrabecular lattice, or perhaps because of the stimulation that such a controversy provides, Keith Porter retains his enthusiasm for research and his excitement about the concepts arising from the study of cells.
Well, I've always theorized and made generalizations beyond the observations, because I thought that was what it was all about. You develop a certain sensitivity after a few years of working with cells. You can anticipate. It's like working many years with an individual. You know what he or she is going to do in response to something that happens, that you say, something like that. You have to be careful not to mention this or that. On the other hand, you know that if you want to delight them, you say the right things. And when you're studying cells, they become persons. They become sort of thing that you can predict what's going to happen, what the story should be.
Lee Peachy and Peter Satir sum up Porter's career so far.
Keith has worked on a number of areas and a number of different problems. So I think you'd have to focus on a more general thing, and I would say cell-fine structure is the accomplishment of Keith's career.
I think the overriding contribution that he's made is what I'd call it is essentially what Lee calls cell-fine structure, but what I would call the organelle doctrine. I decided to make up a new phrase for this. The organelle doctrine says that you can recognize an organelle by its structural characteristics, and that these have consequences for the function of the organelle and for its evolution.
Today, as areas of research and cell biology come ever closer together, both Keith Porter and George Pilati hold firm in their belief that our knowledge of structure is basic in progress toward a comprehensive understanding of cell function.
The cell biology of today started properly with morphology as alpha, okay? But then it moved to this level, and now sometime in the future we'll come back to morphology as omega. Now we are somewhere in the middle of the alphabet.
When we come to get back to time ground zero and begin to think of the cell as a unit, that more and more attention will be given by biochemists and molecular biologists to what is going on in this unit structure. How is it organized? What is the importance of the organization?
He is one of the people who've opened up a whole new world.
Keith was just a good example of how to work, not to work hard and to keep at something until you got it right, until you were really satisfied with it.
I've always been impressed by his eagerness for new ideas, his willingness to break new ground.
His nickname after he left Rockefeller was Papa, because everyone realized that they had a role model that would be hard to match.